I'm Senior Life Sciences Analyst John Vandermosten with the CEO of TuHURA Biosciences, Dr. Jim Bianco. Antibody drug conjugates have been a very well-known asset in the oncology space, and a lot of big deals have been done with them. And specifically, we could think about Pfizer's pickup of Seagen in December of 2023 for $43 billion. They've been valued very highly, and traditionally, ADCs combine a targeting molecule with a toxic payload. But TuHURA is looking ahead to the next generation of this product by doing something new with it. So, Jim, why don't you tell us about how your ADC is going to break new ground?
Sure. I mean, John, you're correct. The first generations were really targeting tumors with toxic payloads. Then investigative sites started looking at whether or not they can take something that targets the tumor and maybe activate the immune system at the same time. Those are termed checkpoint ADCs, but we're not interested in targeting the tumor at all. We're focused on the fact that the tumor lives in this sanctuary where it can escape recognition by your immune system because the cells that surround the tumor essentially secrete various substances that create this protective shield. And so nobody's really looked at, can you shut off that shield and carry with your target in a drug like Keytruda to activate the immune system? So that's kind of what we call bifunctional ADCs. And the first part of it is we've actually identified a receptor called a delta receptor that sits on a cell type. These cells are called MDSCs, and they're the predominant cell that creates this protective shield for tumors. So if we target that receptor, we can shut off that cell's ability to produce that protective shield. And now if we link that drug that targets that receptor to something like Keytruda, it kind of has this double hit phenomenon, right? We can remove the immunosuppression, i.e. the shield around the tumor, and activate the T cells where they currently reside. And that's a totally new approach. It's really a paradigm shift, right? Because we're getting away from the tumor aspect and really focusing on where the tumor lives and how that protects the tumor against drugs like T cell therapies, CAR T, or even checkpoint inhibitors. Why checkpoint inhibitors stop working, right? That environment causes what is termed T cell exhaustion. So if you make that environment essentially more friendly to the immune system, you're exposing a tumor which previously was protected now to the effects of your immune system. And if you can pump up that effect, i.e. with a checkpoint inhibitor, like a drug like Keytruda, or even quite frankly, the VISTA inhibiting antibody that we're looking at. And so we think that this approach is going to start to garner some interest amongst pharma companies that have focused most of their resources on the other side of ADCs, which really is, quite frankly, more like conventional ways to treat cancer as opposed to novel immunotherapeutic ways to treat cancer. And we think we fall into that latter bucket.
Okay. And Jim, I want to get your point of view on the value of ADCs, where you think it comes from. We've identified a number of multi-hundred-million-dollar deals for early stage ADCs that have been picked up in the market. And of course, I mentioned the Pfizer Seagen. Some of these, many of these ADCs are generating multi-billion dollars in annual sales that have been approved. But I want to hear your point of view on where the value comes from and why they kind of outpace other oncology assets in terms of the amount people are willing to pay for them.
Yeah. So, innovations in oncology move the whole field forward. I mean, Keytruda as a single checkpoint inhibitor is doing almost $30 billion a year. But what do you do to make drugs like Keytruda better? And I think that is where the field of ADC technology is now moving. And for example, I mentioned ADC checkpoints. So if you connect a drug like Keytruda, a checkpoint inhibitor, to a drug that's targeting a tumor, you're now having the ability to have that checkpoint inhibitor localized to where the tumor lives. So I think that next phase not only helps protect, quite frankly, the loss of exclusivity on drugs like Keytruda, giving them kind of a renewed life, potentially, more importantly, renewed activity or better activity against tumors. And then similarly with what we're looking at doing is really, again, removing this what's called acquired resistance. It's why drugs like checkpoint inhibitors, or quite frankly, ADC checkpoint inhibitors, stop working because the environment essentially exhausts their ability of those drugs to work. So that's what we think is kind of the next, I don't want to say breakthrough, but the next leap in advancement for these immunotherapies, these cancer immunotherapies. And certainly if pharma companies don't have this in their toolkit, we think that this type of technology, kind of the next, we won't even call it generation because it's really a leap ahead of a generation, quite frankly. I think that this will start to be where ADC technologies move, and I think we'll essentially be leading that pack.
Okay, Jim, and we previously mentioned the delta opioid receptor, which has a lot of touch points throughout the body. I mean, not just in oncology, but through I believe immunity and some other areas as well. Why is this such an important area for you to pursue, and what's the importance of the delta opioid receptor?
Right. So nobody has ever described, so it was TuHURA scientists that first demonstrated that the delta opioid receptor is present in high quantities on immune cells, and that the delta opioid receptor plays a role in immunosuppression. And so by the delta opioid receptor being activated, it actually will allow certain immune cells to become very immunosuppressive. They call these tumor-associated macrophages or MDSCs. And these are the cells that essentially create this soup around a tumor that causes immunotherapy to stop working. So now, for the first time, we've described a central point that we can control potentially these cells in terms of their ability to create that immune suppressing environment around the tumor. And so it's not just novel, but it is really potentially groundbreaking if this technology translates in the clinic to the way we think it will, that if you can remove that immunosuppression in the microenvironment, all these existing drugs that are being developed will work potentially not just better, but longer. I mean, you look at a drug like Keytruda or Opdivo, they work very well when they work. The problem is they stop working. They're not curative therapies. Why do they stop working? Because eventually the environment that they're fighting the tumor in fights it and actually shuts them off and makes them no longer work. Well, that's what we're focused on. And this delta receptor, as we term it, actually can be a very important pivotal piece to removing that so-called environment, that shield, and allowing drugs like Keytruda to keep working, whereas otherwise they would have stopped. And so that's, quite frankly, I think will be very well received when this data is first published or presented in scientific meetings. We hope to have that towards the end of this year at the American Society of Hematology. I think a lot of folks will be surprised that something as simple as an opioid receptor can control an immune response the way it does.